Hi, guys. Let me get everything set up and we'll get going soon. We won't have a video with this on because I am doing this on my iPad today. So you won't see my face when I'm talking. Just FYI, in case you're like, I can't see your face, you're right, is my answer to that. So, but I will, let me really quick get that PowerPoint started and we'll get going here in just a minute. Pull this up on here too.
Hello, all.
Alrighty, let me see. It's been rough. It's been a weird day, guys, so bear with me. Weekly reviews. Slides, downloads, lecture. Okay. PowerPoints. Share my screen for you guys so you can see what I'm talking about. I was hoping I could have the chat open while I was doing this with my other computer that I can't see the chat on my iPad. So I'm hoping to have it open on my other computer, but it's going through updates. Of course it is. So if you have something to say in the chat, just somebody like verbally let me know somebody said something in the chat so that I can open it, because otherwise I can't see it. It doesn't pop up on my screen or anything. I might be able to open it on my phone. I don't want to mess with that right now. I'll wait till my computer comes up. So can you guys at least hear me?
I can.
Okay, awesome. Thank you. All right, so we're going to do our week 4 review.
Yes.
Awesome. Chapter 5, where we'll be talking about the eukaryotes. Chapter 7, which is the viruses, and then our most missed topics from each of those chapters. I was also hoping at the end of this, I don't have a slide for it, but I'm going to try to go over it anyways when I get to it. Those 5 critical thinking questions that I posted, I want to try to go over those and break them down for you guys too, which I think could be helpful. And it might be a waste of your time. I don't know, but I'll go over that at the end. So if you want to stay, you can. If you want to go, go. Okay. So chapter 5, our chapter 5 was going over, let me pull my pen up. Chapter 5 was our eukaryotes. And eukaryotes are our U means true, and karyo means nucleus. So these have a nucleus, which is a membrane-bound organelle, and other membrane-bound organelles. I'll go ahead and list our major organelles. We talked about it in class, but if you're my online students, you might not have gotten that review. So our major organelles are membrane-bound structures that have a specific function within a cell. Our major membrane-bound organelles that we find in eukaryotes are the nucleus, which is where we store the genetic info. Then we have the endoplasmic reticulum, which I'll put as ER, okay, just to save you from having to see me write it all out. The endoplasmic reticulum, we have the rough ER, which is where protein is made, and the smooth ER. The smooth ER is where other molecules like carbohydrates and lipids are made. And we have the Golgi apparatus. The Golgi is our packing and shipping. It can make some modifications and help with folding of proteins and stuff as well, but it's mostly our packing and shipping. So getting things ready to pack and ship them across or out of the cell. We have the mitochondria. which makes energy for the cell. We know it as the powerhouse of the cell. The specific molecule of energy that it makes and is associated with making is ATP. And the mitochondria, ATP is always made through aerobic respiration. This means it depends on oxygen. And we'll review aerobic respiration and all its steps and everything like that in Unit 2. So we're not there yet. You don't need to know all that, but you do need to know that ATP is made in the mitochondria of eukaryotes. Okay. Can bacteria make ATP? Yes. They just don't do it in mitochondria. All right. They do it on their inner membrane, their cytoplasmic membrane. That's another thing that we could talk about that eukaryotes have. Eukaryotes have a cytoplasmic membrane or a cell membrane. Okay. All cells have that. All right. And the purpose of the membrane is to control what goes in and out of the cells. But then if you have internal structures that are also membrane bound, those are our organelles that are unique to eukaryotes. And these ones here are ones that all eukaryote cells have. Some of them may have vacuoles for storing, including storing water or wastes or other products. The vacuoles are what we're storing things in, okay? Lysosomes, which are little compartments for digestive enzymes, for the cell to break things down. Chloroplasts, which are organelles that are important for photosynthesis. So obviously, plants have chloroplasts, okay? Algae have chloroplasts. So those are the only two that we need to know of that have chloroplasts, plants and algae. That's it, not fungi, all right? So keep that in mind. All right, so those are our organelles. The endosymbiotic theory, we could draw a cell. I don't really think it's that important that we draw the cell. I'm not going to have pictures of cells and make you identify things on diagrams. None of my questions ever have diagrams, okay? So you never will be identifying on diagrams in the lecture exams, okay? We might have that in the lab exams, but that's not now. All right, so that's our organelles and their structures. The endosymbiotic theory says about how mitochondria evolved from a cell eating another cell, probably a bacteria, and then that became a permanent symbiotic relationship. Symbiosis means that the two organisms rely on one another for survival. It doesn't just mean that their lives intertwine, but they literally rely on one another for survival. And we think that that's how mitochondria and chloroplasts both came to be. And we think possibly our other organelles may have evolved in a similar way or from that similar process. And the evidence that supports that is that the ribosomes that our mitochondria have are the same kinds of ribosomes that our bacteria have. They also have they have their own genome. Mitochondria are not coded for in your nucleus or in the genes in your nucleus. Mitochondria have their own genes and they multiply on their own, separate from the cell having to multiply. And the genes of your mitochondria are in one big circle, just like we talk about with the bacteria genes, in one big circle. Their chromosome is one big circle. Their ribosomes are the same as bacteria. They also have two membranes. Mitochondria often will draw mitochondria like this, outer membrane, and on the inside we have this big old foldy, but it's all one continuous membrane. So you could think of this as being very similar to an outer membrane and an inner membrane of like a gram-negative organism. We don't have any peptidoglycan or anything like that anymore, but this also suggests the basic shape and organization of like how bacteria have an outer and inner membrane. So that's the idea with the mitochondria. On that inner membrane is where we will be doing our electron transport chain and making our ATP with our aerobic respiration. But we'll review that in chapter 10, just to give you some context though on why we're learning about that, okay? So that's the endosymbiotic theory. Basically, the summation of it is that we think that mitochondria and chloroplasts evolved from bacteria that our cells ate, and it became a permanent relationship over time, evolutionarily. All right. It's not right. Hold on, sorry. All right. All right. So the next thing, we did this, mitosis. You don't need to know a ton about mitosis. I'm not going to go over it a lot. So I'm just going to give you our general overview of mitosis, which is PMAT. Actually, let me. PMAT. All right. So these are the steps of mitosis. You do not need to just name the 4 steps. You should be aware of what happens in each of the 4 steps. So what happens before mitosis even occurs is something called interphase, and it's where we have actually like made a copy of our genome and stuff like that, okay? So we make that copy, but it's in the PMAT phase that we actually start going through the mitosis process. P is prophase. This is our prep step. This is where we are basically that big old huge genome that we have. Your chromosomes do not exist in your cell in tight little neat X's. That's fake. That only happens during mitosis. You also don't have X-shaped chromosomes. You have one chromosome. And whenever it doubles itself, we make a sister chromatid. And they're just a copy of each other. So think of this as your cell, and we've condensed down our copied chromosome, our linear chromosome, we've condensed it down and joined it at the center. And that is what's happening in prophase, is getting all of this stuff ready. Okay, so it's our prep step. All right, we have 2 copies, copy 1 and copy 2 of the same chromosome. They're exactly the same chromosome, 2 copies. Then in the metaphase, this is the one that I say middle. This is where all of our chromosomes will be lining up in the middle of the cell. Okay, and they line up because these spindle fibers attach at the centers of these guys. So this would be metaphase. All right, and we line all those chromosomes up in the middle because what we're about to do in the next part in anaphase, this is apart. So we're going from that and now pulling those apart. So we're pulling that X that was an X in the middle, taking it in its center and pulling each of those sides of the X apart towards the middle. One side of an X, the other side of the X, and pulling it towards the middle. So once we start pulling it apart or pulling it to the other side of the cell. So from them being stacked up and connected in the middle, all aligned in the middle, in the metaphase, in anaphase, we're pulling them apart. In telophase, no. Telophase. I mean, you can think of this, I think of it as the T stands for totally done. But basically you are creating a new nucleus, you're separating the cells, you're dividing the cells apart, you're creating a cleavage between the cells, pinching them off from one another, creating a new nucleus, and getting totally done with the process of mitosis. That's telophase. So understanding what's happening with the chromosomes and with the nucleus in each of these steps, okay? Prophase is preparing. We've made a copy of our genome already. We're just condensing it down and we're degrading our nuclear envelope, that membrane that surrounds the nucleus. Metaphase, we're aligning our chromosomes. Anaphase, we're pulling them apart. And telophase, we're completing everything, budding our cells apart from one another, cleaving the cell down the middle, and recreating our nucleus. So that's mitosis. Mitosis is only for creating an exact copy of the cell. Mitosis is not sexual reproduction. It is just creating the exact copy of the cell. And mitosis is something that only eukaryotes go through. Bacteria divide similarly, but they don't have all these steps of aligning things in the middle and pulling chromosomes apart because they don't have individual chromosomes. Bacteria go through something called binary fission, and it's basically just making a copy of their gene and then splitting the cell apart. That's really it. And it's just a much more watered down version of mitosis. It's also asexual reproduction. Bacteria cannot reproduce sexually. How do we reproduce sexually? It's through a process called meiosis. And I do not cover it much in my class, and I'm not going to test you over meiosis steps, but being aware that meiosis is the process that we go through to create our gametes. And that means our sex cells. Because if we're going through mitosis, MIT, mitosis, we're going from a cell that has 23 pairs, and we're making 2 new cells that each have 23 pairs, or 46 total. That's mitosis. In meiosis, we go from a 23 pair or 46 guy to 2 23 pairs to 23 single, 23 single, 23 single, and 23 single. No pairs anymore. So now we have 4 sets of cells, each of them only 23 chromosomes. So now when you have an egg with 23 coming together with a sperm with 23, the resulting product has 46, which is how much you need to be a functioning human being. That's the idea of meiosis. Like I said, I'm not going to test you on how many chromosomes and where and when and what steps and how it works. Just be aware mitosis creates exact copies of the cell. It's what happens in your body when you cut yourself, when you're growing your organs after you've been conceived and lengthening your bones or whatever. All right. But the sex cells themselves, the gametes, are created through meiosis. So you do need to know that. Okay. All right. I'm gonna erase this because it's just a lot more information than we need. All right, the fungi and their structures. There's 2 basic types of fungi. Fungi, we have the budding yeasts and the molds. I mean, yes, there's mushrooms and stuff too, but for microbiology, there's 2 types of fungi, yeasts and molds. The yeasts bud off of each other, like kind of very asymmetrically. You'll get a new guy kind of budding off of it. And these are by mitosis. And each of these would be a new cell, and they'll eventually grow up to be about the same size. But as they're making new cells, they divide pretty unevenly through mitosis. Molds, however, they sort of branch cells off of one another. And so they'll have one sort of like this, and another one will branch off of that. And then this one might branch off of it, and then whatever. This branching is called, so these are budding yeast. And this is called hyphae. Just branching of cells is called hyphae. Whenever we have a whole mold, like you're looking on that bread, that whole green spot on that bread, we would call that the mycelium of that structure. Generally speaking, it's the whole body, the main body of the mold. And some of our molds, whenever they're ready to reproduce, they'll create these structures on themselves that release spores. Spores, we need to be careful here because endospores that bacteria make are not reproductive, but spores that fungi make are. And so it's just like we've been thinking about, and probably you've heard about with mold spores, we're releasing a ton of spores into the air, quite literally. Mold does that. Bacteria, endospores, do not do that. They do not do that. Endospores do not do that. They are not at all the same thing, okay? But molds do this. And those spores that they're releasing can come in 2 forms also. The 2 forms that the spores can come in are the open ones. The term for the open ones are conidiophores. Conidio is the word that I want you to focus on here. Let me erase this. And I'm going to zoom in a little bit so I can write this more clearly. My pen. Come on. Don't fight me here. Does not want to let me zoom in. I don't know why. Here we go. Here we go. Here we go. Okay. Open, like this. The term for it is conidio. Organisms that make the open spore-forming structures are called conidiophores. These structures themselves are called conidiospores. And like I said, I'm not going to make that distinction on the test. Definitely know conidio is the open form of the spores. And some of them have ones that they'll make that look like the same, but they have a big covering on the outside. The covered ones are sporangio. Again, the organisms are called sporangiophores. This structure that is covered, we call it a sporangiospore. All right. But if you know sporangio are the covered ones, conidio are the open ones for the molds and the asexual reproduction process of mold. Okay. Next are our protista. Protista includes both the algae and the protozoa. Algae are either multicellular or single cell. When we start thinking about our plankton, plankton often fall into either algae or protozoa. They can be one or the other. If they are photosynthetic, they're going to be algae. All right. But algae can be single or multicell. Sea kelp is algae. It's a multicellular algae. Algae don't have true tissues. They kind of look like they have different parts going on, but there's no clear defined tissues. The tissues aren't really different from one another in their arrangements and their compositions. So algae can be multicellular or single cell, but they don't have tissues. Protozoa are always single cell organisms. All right. We had 4 ways to categorize protozoa based on their motility. We had the ones that move by flagella. We had the ones that move by cilia. So remember flagella, if you were a protozoa that looked like this, your flagella would look like that, right? Big old long flagella. Cilia, you might be a protozoa that looks like this, and you have all these little oar-like appendages that sort of beat the liquid around you. They're tinier. They're much, much tinier than flagella. They can be used for feeding, but also can be used for motility. You can have pseudopods. Pseudopodia is the better scientific term, or fake feet. These are also known as our amoebas. All right, so they literally are like a blob of a cell that sticks out a blob, and then that blob pulls the rest of that cell toward it. That's how amoebas are motile. And then our last group are known as the sporozoa. And the apicomplexans are in this group. And I won't make you type out apicomplexan as an answer, but you should be able to identify that apicomplexans are the sporozoa. They are non-motile, not truly motile, not truly motile. So those are the 4 types of motility that you can have in protozoa. All right. All right. Protozoa can also basically switch. Some of them are capable of switching between dormant. Sorry, I'm trying to center this. Stop fighting me. The dormant and the active forms. And so we remember this from bacteria because we talked about this with endospores, which I just mentioned just a second ago. I forget when I zoom in, it doesn't let me erase. Great. I didn't mean to do that. Hold on real quick. There it goes. Okay. Yeah, let's just go to the new slide. Why not do that? Okay. So with our protozoa, we can switch between the dormant or inactive form. I don't like how I'm writing this. I wish I could move this because now it's weird. It's weirdly positioned, but that's fine, I guess. Dormant or inactive versions of cells versus the active multiplying, infection causing, toxin producing, sorry that the P went off the screen there, but you get the idea. All right, protozoa that do that versus bacteria. Like I said, if I could move it over, I would, but I can't on this. All right. So protozoa that are dormant and inactive, we call those cysts. Bacteria that are dormant or inactive, we call those endospores. All right. Again, non-reproductive. Never for any of these two is this reproductive. One cell begets one cyst. One active cell begets one endospore. It's not reproductive, it's just protective, okay? The active metabolic guy of the protozoa is known as the trophozoite. In bacteria, it is known as the vegetative version, all right? In bacteria, the ones that can switch are either Bacillus species or Clostridium. If you think you're not going to get tested over that, you're fooling yourself. I think I mentioned it enough. I've made it pretty clear. I'm going to make you use that knowledge at least once on the exam, okay? So you do need to know Bacillus species can make endospores. Clostridium species can make endospores. No other species of bacteria can make endospores. Bacillus and Clostridium species, you do need to know, okay? You do need to know that both of these are gram-positive bacteria. Now, when we're talking about a protozoa, which ones can make cysts and trophozoites? There's way too many. I'm not going to make you recall which ones can do that. I mean, there's way, way, way too many. Most of them do it. Not all of them do it, okay? But most of them do. For example, malaria does not do that. Malaria is only basically the trophozoite version. There's no cyst of malaria. But there's a lot that do switch, okay? When I'm talking to you about, okay, the bacteria that do this, the bacillus or the clostridium, and those will be in the name somewhere. Even lactobacillus, that's in the name, bacillus is in the name, those are endospore formers, okay? And if it has bacillus or clostridium in the name, they are endospore formers and they are gram-positive, okay? What about our protozoa? Are they gram-positive? The answer is no. The answer is a hard no, because gram stain only tests basically bacteria for the presence or absence of a thick peptidoglycan wall. Protozoa do not have a peptidoglycan wall, so they cannot be gram positive or gram negative. That's a term that only applies to bacteria. Okay, any questions about that? Didn't mean to click that. Sorry, it was written on the side of the screen like that. All right, our next ones, well, we'll just use the other side. We'll use a new one for, forget that it deletes that stuff. Okay, so our next part is our helminths, which is our last part. Helminths are our parasitic worms. So these fall into kingdom Animalia. These are animals. These are multicellular, always, with true tissues. And they even have like rudimentary digestive tracts, mouth parts, and things like that, right? So these are multicellular organisms. They have true tissues as part of their existence, okay? For our helminths, you are either a roundworm. The term for our roundworms is nematoda or nematoda. For our flatworms, the term is platyhelminthes. There are two subgroups of our flatworms. All right. These are going to be our tapeworms, which are exactly like what they sound like. They're long worms that are flattened. They may have segments like we think of with traditional tapeworms and how the ends of them can break off. Those ends, when they break off, are called proglottids. They contain their eggs inside of those segments. Okay, so that's tapeworms. The other flatworms that we know are flukes, which are much smaller. And we think of planaria and stuff like that. These guys are very small. They look like flattened, like, I don't know, like leeches almost, but they're flat. Okay. And these guys are very, very small. They're microscopic. Okay. All right. So our tapeworms are known as cestoda. That's our fancy word for them. And our flukes are known as trematoda. All right. So that's our basics for our eukaryotes. Do we have any questions about our eukaryotes before I move on? Okay. Not what I'm trying to do. There we go. All right. Reviewing chapter 7. Viruses are obligate intracellular parasites. And so we talked about this in class, but to review that really quick. Obligate means you're required to do it. I mean required. Not optional or you choose to. I mean you are required. You must be. Intracellular. This is one word, but the intra part means inside. And cellular obviously is cell, inside of cells. And parasite. This means that the organism harms a host and the relationship is required for survival. This is a type of symbiotic relationship. Many of us think of symbiosis as being beneficial. That is an incorrect understanding of that term. We'll talk more about those terms in Unit 2, but symbiosis basically means that you'll have a relationship between two organisms that is required for survival. And in a parasitic symbiotic relationship means that one organism is feeding off of another one, causing harm to the one that it's feeding off of. So one organism benefits and the other one is harmed. The one that is harmed, we call the host. The one that is harming, we call a parasite. We think of ticks and leeches as being common examples of parasites. Viruses are parasites. They are basically using our cells' resources and machinery for their benefit while harming the host, which is us. Okay. So all viruses are obligate intracellular parasites. They cannot multiply without being inside of a cell. They rely entirely on that cell's machinery in order to exist. So here's our basics of our virus. There's really just two parts that are required. And there's several optional aspects, and one of them is a pretty major one that we need to know. Okay. So for our viruses, all viruses have a capsid. This is a protein coat surrounding the nucleic acid genome. All right. The other thing that all viruses have to have, I don't know if I want to make it that big. No, 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 no. All viruses have two things. Acid. Just the protein coat. Surrounding nucleic acid genome. And obviously the second part is the nucleic acid genome. Just trying to get there. Their genome. They do have genes. The genes are very basic. They might even just have like 2 genes. Okay. Some of them have like 20 or 30 or even 100 genes. It just depends on the virus and what its parts are. Might have different kinds of proteins that come together to make up the capsid and whatever. But their genome is nucleic acid. Notice how I didn't call it DNA. Our genes, all cells, have DNA as their genome. All cells, bacteria, archaea, and eukarya, those domains that we talked about for our cellular organisms, all 3 domains have double-stranded DNA helix like we're used to thinking about. Viruses are not cells. This is important. If you ever have a question on a test or on a Kahoot or anywhere, and it starts talking about viruses and their cells, you know immediately that's not the answer. Viruses don't have cells ever. Okay, so they're acellular. They're just these 2 things. And maybe some viruses have an envelope. And that isn't all. They can have other things too. Maybe they have certain enzymes that they carry with them, like reverse transcriptase, which is a special enzyme that HIV has, which it won't make sense to you why I'm talking about it until we get into Unit 2, but we will talk about it. They have special enzymes that they can carry, certain toxins that can be associated with them. They might have different kinds of proteins that are going to be on their surface that have different functions, like flu. When we talk about flu and we talk about H1N1 or H5N5 or whatever, the H stands for hemagglutinin and the N stands for neuraminidase. And those are special proteins on the surface of the virus, and each of them does different things. So some viruses are more complex than others. Okay. These are the basics of them. Some of them have envelopes and other enzymes or whatever they might have. I'm not going to talk much about the enzymes and whatever in this chapter. We're just getting our basics down. Many of them have an envelope, though, not all of them. If you have an envelope, we call you an enveloped virus. And the envelope, what it is membrane stolen from the host cell. Some viruses, if you are a DNA-based virus, you multiply in the nucleus because that's where DNA stuff happens. If you're an RNA-based virus, you might multiply out in the cytoplasm or in the nucleus. It just depends on the virus. If you are a DNA virus and you multiplied inside of the nucleus, you might actually push out through the nuclear membrane, not the cell membrane. So when I say it's a membrane stolen from the host cell, it could be the nuclear membrane or it could be the cell membrane, the cytoplasmic membrane. It's either one, but it's a membrane and it got stolen from the host cell. We stole it by just pushing through it. We call it budding. It's different than yeast's budding. In this case, viral budding is whenever the capsid and the genome literally just push through the cell membrane, the cell's membrane, whether it's the nuclear or the outer membrane. They just push through it and then just blanket themselves with that membrane there. And then they pinch it off behind them to leave behind those enveloped virus particles. A single active viral particle, we call it a virion. Okay. All right. And again, Don't think that we're ever done with viruses. We're not like moving on forever with viruses. We're definitely coming back to them later on. Many diseases are caused by viruses, and the way that our immune system responds to viruses is drastically different than how it responds to bacterial infections. And so it's really important that we understand their distinction and how they function so that we can understand that stuff later on in the course. Okay. After Unit 1, we will temporarily talk about viruses in our genetics chapter, but then pretty much beyond that, we won't pick them up again until we get into immunology, I'll be honest. And then, of course, we'll talk about them extensively in Unit 4 when we talk about our diseases, just to put into perspective about this, okay? All right, the capsid, our protein coat, it's made of parts, made of subunits called capsomeres. Capsomeres come together to create the capsid. All right. You can draw our naked viruses. If you're just these two and no envelope, we call you a naked virus. Okay. If you have an envelope, we call you that enveloped virus. All right. So there's two types of viruses there. Okay. All right. You have two types of capsid structures. We have the icosahedral. It's made up of triangles that are stacked up onto each other like a 20-sided die. Okay. Just 20 triangles stacked up on each other to make a big structure. Or you are helical. And if you were a genome, let's say this is the genome, the helical capsid just wraps itself around in a helical fashion and just goes around it like this. Just imagine that those little capsomeres are building around and around and around and wrapping around that nucleic acid. That's how the helical capsid works. Whereas the icosahedral one, they stack up and look more like that. You'll see it's drawn as a hexagon, but just imagine all the different faces in a three-dimensional set stacked around that genome in the middle. Okay, so it's our two types of capsids. The types of genomes that you can have, we know that they're nucleic acid in nature, but you are either DNA or RNA. You're not just DNA. Your DNA could even just be single-stranded, but you could be double-stranded like a normal cell has. But the RNA could be double-stranded. Nowhere in our own cells do you have double-stranded RNA. Eukaryotes, bacteria, do not have double-stranded RNA. So that's wildly divergent from what we're used to seeing in biology. But you could have that in a virus. Or it could be single-stranded. The single-stranded guys for both DNA or RNA can be positive sense. This is a plus. Or negative sense. If you're positive sense, then that means that we can take whatever your sequence is, A, T, C, and then turn that into protein directly by reading with a ribosome. If you're negative sense, it means we have to take your A, T, C, turn it into T, A, G, and then that can be made into protein. You guys remember transcription and translation, right? So this is just understanding the gist of that. I'm not going to test you on the specifics of that, but I want you to have a sense of what it means to be positive or negative sense, because in Unit 2, that's going to carry. I'm not going to test you on what positive and negative sense mean on this test, but you'll need to understand it for Unit 2. So that's why I'm bringing that up. But you will need to know that viral genomes could be DNA, double-stranded, single-stranded. If it's single-stranded, it could be positive sense or negative sense. Even though I might not make you explain what positive sense means, you know it could be positive sense or negative sense if it's single-stranded. I hope we're tracking with that. Okay. So this is the gist of all viruses and their setup. Okay. Any questions about this stuff that I've drawn? Because I'm going to go more on to the viruses here in a second, but once I switch away from this screen, this is all going to disappear. This doesn't save. Okay, so I'm going to go back here. We've covered this and this. Spikes. Let's say we have a virus that looks like this. This is a naked virus. We might have a virus that looks like this. This is an enveloped virus. I haven't drawn it completely. Here's your envelope. Got the membrane on the outside. All right, so those are two different types of viruses. Spikes are special proteins on the outside of the capsid or on the outside of the envelope. They must be on the outside. If you have an envelope, your envelope is on the outside, okay? So whatever your outside layer is, depending on what type of virus you are, you will have spikes. These are glycoproteins. Remember, glyco means sugar. Proteins are proteins. They're amino acids linked together, okay? And these work as keys to help access the host cells. And so they'll bind with certain molecules on the surface of host cells. And when they bind, it causes the cell to either eat up the virus or causes the virus to merge with the host cell membrane. It just depends on what type of virus you are and how your glycoprotein works. And so when that happens, that allows the virus to get into the cell. So your spike proteins determine your host range, as well as your tissue range, what cells you can infect and what species you can infect. So those are the spikes. Viral life cycle, and you need to know these steps and what they mean. Adsorption is attaching to the host somehow. Your target cell, when your spike interacts with whatever molecule you interact with. If you're COVID, if you are SARS-CoV-2 virus, your spike protein interacts with an ACE2 protein on your host cell. When that interaction happens, that's adsorption, okay? Penetration is whenever you enter your host cell. So our spike bound and the cell ate us up as a result of binding, or our surface merged with the host cell. So we could see this being the host cell membrane. Continuous. A cell comes and a virus comes and docks. This would be adsorption. Here's your virus, there's its spike, whatever. And then with our penetration, this might then cause the virus membrane to merge with the host membrane. And then dump whatever was inside into the host cell as a result of that. So that can happen, or the cell can eat it up. There's all sorts of different ways we can get in. Okay, so that's the penetration step. Penetration then almost immediately leads to uncoating. If we're dumping our contents in there, this is part of the uncoating step as well. If we have to get eaten up first, then our digestive juices will help with the uncoating. Okay, so this is removing the envelope and the capsid. Then comes synthesis. This is making copies of all the virus parts, including the genome and the capsule mirrors. That'll eventually be the capsid. And then we have, of course, assembly, building the new virions. And then release. Either the cell, you'll build up all these viruses inside of your cell, and your cell might actually like just be like, what is all this junk? Put them in vesicles like in the Golgi apparatus would do. Remember, that's our packing and shipping. Golgi will pack and ship those virus particles and say, get out of the cell, and just dump all those viruses out of the cell. But where do those complete viruses want to be? Outside the cell, so they can go into the next cell and cause a new infection, right? So that can happen. Or we can have that budding thing, which is the reverse of this image. taking that capsid, pushing it out of the host membrane, and covering ourselves with it. All of the effects of making these virus particles can lead to cytopathic effects. These are changes that we would see at the microscopic level, the light microscope level of our cells. Them no longer attaching to surfaces, them merging with each other, them having issues with adherence or shape, or them having inclusion bodies with too much protein production while we're building up capsids and things like that. Those are all visible in microscopes and are bad effects that are caused in our cells as a result of viral infections, and those are cytopathic effects. Cyto means cell and pathy means disease. And so those are all visible with microscopes and are effects of a viral infection that you can see as the viral infection progresses and makes new viruses inside of the cells. All right. All right, so we're going to talk about bacteriophages next. I'm going to make a whole nother slide with our bacteriophages as we talk about the lytic and the lysogenic and stuff like that. All right. Let's see. All right. Bacteriophages. These are viruses that infect bacteria. All right. We also call them phages. And you'll often see me abbreviate it in some of my lectures and stuff like this. This is phi, which I use to abbreviate phage. So when I start talking about macrophages, which are not viruses that infect bacteria, they're a white blood cell that you have. But macrophages, I often abbreviate as M-phage. Okay, so that we can track that later on. But anyways, so these are, oh no, I clicked off of it. I didn't mean to. Viruses that infect bacteria. Remember, viruses can infect any type of cell. That includes any cell that falls into any of those domains, eukarya, bacteria, or archaea. So remember, our prokaryotes or eukaryotes are our bacteria and our archaea. Those are still cellular organisms. They can be infected by viruses. There we go. Okay. All right. So there's a couple there's a couple things we need to know about bacteriophages. Number one, they only infect one type of bacterial species. What I mean is one type of bacteriophage only infects one type of bacterial species. So if you are a T4 phage, you only infect E. coli. But there are other phages that only infect salmonella. There are other phages that only infect streptococcus, whatever. Okay. So they only infect one bacteria species. This is important to remember for later when we get into Unit 3, we'll reuse this information, okay? But phages only infect one bacteria species. Number 2, they multiply until they burst the cell open. Luckily, our viruses don't do that to our cells. It's incredibly rare to see in our cells, but in bacteria, this is how they always function. That process of multiplying until you burst that cell open is known as lysis. Okay. Number 3, they always inject their genome into the bacterial cell. So like if you have a bacterial cell and that phage comes along, it'll land on top of that bacterial cell and then it'll squat down with its legs bent up like this and inject the genome into the bacteria. Okay. Phages that switch between that typical lysis cycle replication cycle, and switching from that to incorporating their genes into the host cell. Phages that can do that are known as temperate phages. All right, so the two life cycles of the temperate phages. The first life cycle is the lytic phase. This is that replication and bursting phase. Also known as lysis, right? The other phase is known as the lysogenic phase. This is I focus. I know that they both sound like they have lysis in the name, but this one has gene in the name. And so the lysogenic is the gene name, and that's lysogenic is where we're going to be seeing the virus genes. No, let's not mess it up this time. Insert into the bacteria genome. Okay. So I can switch. In human beings, we can have this happen too. When this happens in eukaryotes, we call this a latent infection. Oh no, I can't draw right underneath that. Infection, okay? When the virus genes that are being inserted are in a phage, we call this then the prophage. When this happens in eukaryotes, we call this a provirus. Examples of provirus situations in humans would be in HIV. In HIV, the HIV virus inserts its genes into the genetic information of our T cells. And that is a permanent insertion. And then when that happens, that is now a provirus inserted into your T cell genome. Okay. In bacteria, we call that a prophage. So there is a difference between those terms. Okay. When we have insertion of viral genetic information into our genes, it can sometimes interrupt our gene cycle. When that happens, that interrupts our genes and how they replicate and things like that. That can interrupt our cell cycle too. And that can lead to over replication of those cells, and that is cancer. When that happens, that's called transformation. And we call viruses that can cause transformation, we call them oncoviruses. Also known as cancer-causing viruses, those are oncoviruses. Oncoviruses cause cancer. When cancer is caused by viruses inserting their genes into our genome, we call that process transformation, to clarify that. Okay. All right. Sometimes, whenever we have viruses inserting their genes into the bacterial genome, we might get new traits. If we get new traits, we call that lysogenic conversion. I had a student ask me about one of the quiz questions, because one of the quiz questions said something about it gave new beneficial traits. The traits are beneficial to the bacteria. It's not beneficial to you as a person getting infected with that bacteria that got infected with the virus that gave the bacteria new genes, but it's very beneficial for the bacteria because now the bacteria can make these toxins that help them cause disease. Any gene that helps a pathogen cause disease, we call that a virulence factor. We've mentioned it previously, and it will come up multiple times throughout the course. But anytime you get a new virulence factor, which is something that helps you cause disease from being infected by a virus, okay, that is lysogenic conversion. The process of being lysogenically converted to have your new genes because you got infected by a virus, that through lysogeny, that process is called transduction. We've given you new genes through a viral infection. That process is called transduction. Okay. All right. Questions about this stuff? Because now is the time. We're going to go into our questions next, like our example questions. All right. Proviruses. Okay. The last thing in this chapter were our prions. Prions are misfolded proteins, and they, instead of being folded like this, they're now folded like, I don't know, like this. And they come near the normal protein that should be folded like this and cause them to fold like that instead. When they're misfolded, you don't perform your function correctly, and you also don't interact with your environment correctly. Okay, so two bad things. You're not performing your function, you're not interacting with the environment correctly. And what that usually means is that these misfolded proteins accumulate with each other, and then they cause holes in the brain. This disease that results from that is called spongiform encephalopathy, which I know that we mentioned it previously, but I'm restating it again. Spongiform because it creates holes in the brain, like a sponge has holes. Encephalo means brain. Pathy means disease. So that is what results from these prions or prions. I don't care how you pronounce the word, okay? But just definitely be able to recognize it and even potentially spell it on the test correctly. The test does grade on spelling, okay? All right. So that's all of chapter 7, I think. That's all you'll need to know from chapter 7. I had some students ask me about what about the virions, not virions, what are they called? The viroid particles. So those are RNA molecules that are just naked RNA molecules, and they can cause infection in plants. I don't have them in my learning objectives, and so you will not be tested on them, and so you don't need to know them. If it's not in my learning objectives, and I didn't cover it in the lecture recording, then I will not test it. I will not test you over it. It has to be both in your learning objectives and covered in my lecture recordings. If it's not both of those things, I absolutely will not test you over it. I do not test out of my textbook outside of that. So FYI. Okay. Here's our most commonly missed topics for our eukaryotes. Knowing our organelles. This is a pretty good little review of some of those guys we just covered. The nucleus contains your genetic information. The endoplasmic reticulum, the rough ER, is where we have protein made, and the smooth ER is where we're making anything that's not a protein. Okay, your carbohydrates and your lipids. The Golgi apparatus is our packing and shipping facility. The mitochondria is the powerhouse where we're making ATP, our energy molecule of the cell. And then chloroplasts are the site of photosynthesis in plants and algae. When we go through photosynthesis, we produce oxygen. In our questions, in a laboratory experiment, a eukaryotic cell, so we know it has a membrane-bound nucleus and membrane-bound organelles, we expose it to a chemical that disrupts the integrity of the mitochondrial membrane. What's the mitochondria? It's the powerhouse. It makes ATP. After exposure to this chemical that disrupts the membrane of the mitochondria, we have a significant drop in ATP and increased oxidative stress. This means that we are now having a buildup of molecules that are involved in electrons exchange. Okay. We'll talk about oxidation and things like that more in chapter 10. We'll review that stuff, but I know you talked about it in chemistry. We should have an idea of what it is. So which of the following cellular functions is most likely impaired as a result of, damaging our powerhouse, our ATP producer? Well, we have issues with folding proteins and modifying them. That's associated with the endoplasmic reticulum, shouldn't be affected. Intracellular digestion by lysosomes shouldn't be affected. Energy production through aerobic respiration in the mitochondria, yes. Synthesis of lipids and steroids in the smooth ER, no. This question is pretty straightforward, but again, we got a lot of words in here. Sometimes for some of you guys, just picking out from the words what the question is can be difficult. So we need to be able to have some understanding of the concepts in order to pick out what the question is actually asking. This literally gave us the answer in the question. So it's just a matter of breaking down the words, right? A research team is studying a eukaryotic cell that shows a decrease in protein synthesis. Where do we synthesize proteins? The rough ER. Okay. They observe that the cell's rough ER, okay, easy enough, appears significantly damaged. So we've got damage to our protein producing organelle. And its ribosomal RNA levels are low. Ribosomal RNA, our RNA, are what make up our ribosomes, which are what make protein for us. Which of the following conclusions is most accurate regarding the impact on cellular functions? Do you have a decrease in protein synthesis because of a malfunction in the Golgi apparatus? No, the Golgi is not important for producing the proteins. It's important for storing or transporting them. The rough packaging and transporting them. The damaged rough ER and low ribosomal RNA levels suggest that protein synthesis is impaired because the rough ER is where ribosomes attach and we have protein synthesis. It's exactly what's going on. Malfunction of mitochondria. No, that's where we make ATP. That is separate from this. Now, I'm going to stop really quick. If you damage your mitochondria and you cannot make ATP, can you still make protein? Hard no. You need ATP to do things in the cell. Protein is a building process. And if you were building a building, if you were building a house, would you need energy? Yes, and your cells do too. Anytime they're building something, they need energy. And the energy source for cells is ATP. But that's not a direct connection, right? Okay. The reduced activity of the lysosomes. Lysosomes are important for digesting things in the cell. That's not what's being affected here. All right, chapter 5, next concept, trophozoites and cysts. Remember, trophozoites are active, motile, feeding. They're the ones that are causing infection. They're the ones making toxins. Cysts are dormant and resistant. They are inert. They're adapted for harsh conditions, just like endospores. There is more information on the topic here if you would like to review it more in greater detail. We're going to make our little cheat sheet notes out to the side. In protozoa and in bacteria. Why not just always write this down on your cheat sheets, on your scratch paper? Active and dormant. The active guys are for reproducing, causing disease. The dormant guys are for getting into the body, but these are the guys that are resistant. Okay, protozoa, our multiply active guys are the trophozoites. Our resistant and dormant guys are the cysts. For bacteria, vegetated and endospore. Just to recall, just to remind you guys, we can't repeat it enough. All right. A parasitology lab is examining a patient's stool sample and identifies both trophozoites and cysts of a protozoa. Okay. Remember, the trophozoite is the multiplying version. The cysts are the dormant, but they're used for transmission. A patient is experiencing GI symptoms, diarrhea, and abdominal pain. Based on the presence of both of these life stages, which of the following statements best explains the roles of each of those life stages in the infection cycle? Trophozoites are responsible for causing symptoms in the host due to their active feeding. Okay, trophozoites are active and they are the feeding state. Cysts help with transmission. They help get from an area where they had to be dormant and resistant, but they help getting into the body. So this is probably our choice, but never do that. Okay, can we agree on this? Read through all of your choices before you solidify your other choice, okay? Just make sure that there isn't a better answer. Trophozoites are primarily involved in survival. No, that is the cysts. So it's not B because that is backwards. Trophozoites and cysts both contribute equally to infection symptoms. No, cysts cannot make toxins. They're not multiplying. They're not causing infection. Only trophozoites are. Cysts are only for getting into the body. Same thing with endospores. All right, next. Trophozoites are dormant. No, that's cysts. Okay, so that's why we're going to stick with A. Make sure you break it all down. In an experimental study, researchers exposed protozoan cysts, dormant. Just go ahead and make our little note here. I don't know if it's relevant. Dormant and resistant. Not multiplying, not active, not causing infection. We expose it to heat and desiccation. Okay. The study finds that the cysts survive these conditions and the trophozoites do not. What can be concluded about the function and adaptation of protozoan cysts? Questions like this are very common on the test. This is not asking a lot. It's not. It's just wordy. So we got to break it down and figure out what it's asking in relation to our general understanding. All right, so cysts are adapted for active growth. No, trophozoites are. Cysts are designed for survival under adverse environmental conditions. Yes. Making them resistant to heat and desiccation. Yes. Whereas trophozoites are less resilient. Yes. Probably B, but let's see if there's any better answer. Cysts are more susceptible to environmental stressors. No, they're the resistant ones. Cysts are involved in active reproduction. No, that's the trophozoites. So we're sticking with B. I didn't even make it through those other choices. You see, I didn't even read all of the other choices, but part of those choices were wrong. We can rule them out. Okay. For our viral questions, cytopathic effects. Remember, cytopathic effects are basically structural and functional changes to the host cells that happen as a result of viral infection. They happen because the virus takes over the cell's machinery to replicate itself. We can see cell lysis, bursting open of our cells. We can see cells swelling or growing. We can see syncytium formation. This is whenever, if you're a big old cell, and let's say your skin cell. I mean, your skin cells are very close to one another, right? Or your liver cells. You got a whole bunch of liver cells all next to each other. So these are your cells. And inside you have viruses growing in them, right? And on the surface, we've already said some of those viruses will want to push out and cover themselves with your membrane like that, right? So in order to make that work, the viruses stud the surface of our cells with the spike proteins so they can be covered with their spike proteins when they get out. But the spike proteins are key to get into cells. So sometimes this causes cells to merge together. So instead of being cells that were touching each other, they're now cells that will merge together like this. And this union is called a syncytium. Syncytia means multiple. Okay. So when we talk about RSV, which is respiratory syncytial virus, RSV causes these to happen. Okay. Inclusion bodies. This is whenever you have a ton of these proteins being made inside of the cell, and they'll sort of just like crystallize on top of one another in these big arrays of protein, 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 protein. These are inclusion bodies. Accumulate viral components in the cell. Okay, and we can see these in microscopes, just to clarify. So cytopathic effects are changes in cell structure and function caused by viral infection. I know we're going a little bit over time from what I usually attribute this to, but we have a hard time usually with the viruses, so I want to take as much time as I can to cover them, okay? So this is cytopathic. And we got an exam coming up, so I don't want to leave anything out. All right, which of the following best describes a cytopathic effect in the context of a viral infection? the viral protein that inhibits host cell replication. Was that something that was relating to the structure and function caused by the viral infection? That is not something that we will actually see. These need to be like visible in a microscope. Okay. So we can't really see that the cell isn't replicating as a result of that. That's not really something you can see, but you can see cells getting bigger, bursting, forming, merging with one another, or large bodies of protein in there. Okay. So that's not a great answer. So let's see if there's better ones. Structural and functional. Okay, well, this is literally our definition that we wrote up there. So it's going to be this one. Process by which viruses are released from infected cells. This is not a cytopathic effect, it's part of the life cycle of the virus. The ability of viruses to integrate into the host cell genome. This can cause problems with the cell, but not cytopathic effects, really. Okay, so B is our best answer for this one. These are all possible changes we can see in the cell, but our best answer for cytopathic effects is B. A laboratory technician observes that cells in a culture have fused to form multinucleated giant cells. This is called syncytia and exhibits severe cytoplasmic swelling. These are cytopathic effects, right? When we start seeing cells merge and forming that syncytia, that's what we're looking for. So based on the names of some viruses, which of these do you think is associated? Herpes simplex, influenza, human immunodeficiency virus, or respiratory syncytial virus? I'm not ruling out that these other viruses could do that, but based on the name alone, we know that this happens. Syncytia happens with RSV. So that's our answer. All right, temperate phages that can switch between the lytic cycle and the lysogenic cycle. Okay, so this is a lot of information about these types of cycles leading up to our definition of lysogenic conversion. I'm going to make our little cheat sheet notes to answer our questions for now. But if you want to read more about these processes, it is here on this slide. All right, so for now, temperate phages switch between lytic, which is our multiply and burst phase. Bursting is also known as lysis. And alternating between these two, lytic and lysogenic, which is whenever the virus genome, the phage genome, inserts into bacterial genome. All right, if this gets new traits, if new traits for the bacteria, then this is called lysogenic conversion. Okay. All right, which of the following best describes the main difference between lytic and lysogenic cycles of temperate phages? Again, I focus on the gen part. The gen is when you insert the genes. So the lytic cycle involves integration of phage DNA into the host genome. No, that's lysogenic. The lytic cycle results in the destruction of the host cell through lysis. Okay, we got that. While the lysogenic cycle involves integration of a phage DNA into the host genome without immediate cell death. Yes, that can lead to lasting incorporation. The lytic cycle occurs in all types of bacteria, while the lysogenic cycle only occurs in gram-positive bacteria. That's nothing. At any point did we talk about that being unique to gram-positives or anything like that. So don't pull new information out when your lack of understanding is what's holding you back there. Okay, so we know B is correct. Don't let something like this fool you. Well, I don't remember her talking about that. Could that be it? You know B is correct. Know that B is correct and lean into that. Okay. Lysogenic cycle produces new phage particles immediately. No, this is the incorporation and hiding out in the genome. The lytic cycle is the one that produces the phage particles immediately. So B is still our best answer. A patient presents with a bacterial infection and caused by a strain of Corynebacterium diphtheriae. This causes a disease called diphtheria, which you've probably not heard of in a while, but it is part of your Tdap vaccine profile, which is tetanus, diphtheria, and pertussis. Okay. So we don't have this here, but it is a respiratory disease, and it is caused by the toxin, diphtheria toxin. Based on what we have learned about lysogeny, which of the following explains why this strain can produce the toxin? As it turns out, relating to lysogeny, you can get toxins from that lysogenic conversion situation. The phage incorporates into the bacterial genome and carries with it genes to form diphtheria toxin. So that's what's happening. That's what this is getting to. We need to know, you need to get to that thinking, oh, you can make toxin because it's a new gene introduced by lysogenic conversion. And that's why she's asking about lysogeny here. So the bacterial strain is carrying a prophage that has integrated into its genome, which encodes the toxin. This is literally a better wording of what I was trying to explain. So it's probably A. The bacteria has a high mutation rate that causes it to produce toxins. Mutations might lead to development of new toxins, but they can also lead to death of the cells. Mutations are mutations, okay? But that's not relating to lysogeny. I'm asking you to relate it specifically to lysogeny. The bacteria has a high resistance to antibiotics that leads to toxin production. This is not a thing. The infection has caused the bacteria to undergo genetic recombination, leading to toxin production. Now, is lysogenic conversion caused by transduction? Yes. So transduction is whenever you have this whole situation happening with your temperate phage, leading to lysogenic situation, bringing in these new genes. That is transduction, and transduction is a type of genetic recombination. But genetic recombination isn't just transduction. There's a lot of things that can fall under the category of genetic recombination. So infection has caused bacteria to undergo genetic recombination, leading to toxin production. It's not wrong, but the more accurate answer is A. This is what I'm meaning by these questions are a little tougher than what you're going to see on the test, but you'll see some tests that will require this kind of critical thinking because we've got some answers here. This is not wrong. D isn't wrong, but A is definitely the most correct answer. Okay? Because we're talking about specifically lysogeny. Okay. Any questions about chapters 5 and 7? Or any general questions? Because I'm going to go into our critical thinking questions next. We good? Okay. Let's see. How do I want to do this? Let's open it up in here. Let's go to announcements. We had our critical thinking questions for exam 1. All right. Again, I encourage you guys to work through these and see if you can get to these answers on your own, but I'm going to go ahead and try to break them down where I can, okay? I'm trying to find my thing that lets me draw on the screen because that would be helpful, but I can't. So I can't draw on the screen to address this. Hold on. Let's copy and paste it. Let's put it in GoodNotes. Okay. All right, so I've copied and pasted this into GoodNotes so that I can write on it for you guys, all right? All right. Patient of yours has an unknown bacterial blood infection, also known as septicemia. Okay, so the word for a blood infection, we call that septicemia. Bacterial blood infection, septicemia. We perform a gram stain on a blood culture, and we show a mixture of gram-positive and gram-negative rod-shaped cells. They're both rod-shaped. Do you guys remember the term for rod-shaped bacilli? Just recalling that. What answer choice best describes what is going on in your patient's blood cultures? The patient is infected with a species of bacterium that can alternate between gram-positive and gram-negative. Can bacteria alternate between gram-positive and gram-negative? No, that's not a thing. So this is A on the actual thing over there. C, D. So it's not A. Okay, they can't alternate. You either are gram-positive with a thick wall, or you're gram-negative with a thin wall and an outer membrane. You cannot switch between the two. You are one or the other. So that's not our answer. The patient's blood culture contains 2 bacterial species. One of those species is gram-positive, and the other is gram-negative. Can you guys think of any reason why this might not be the answer? Because I'm thinking probably so far this sounds more likely. C. This result is not possible because there's no way for any culture to harbor both gram-positive and gram-negative bacteria simultaneously. I would hope that you remember that we had cultures in lab that had both gram-positive and gram-negative, and you'd use that common sense memory to rule that one out. The lab likely skipped the alcohol step in the gram staining procedure. If you skip the alcohol step in a gram stain procedure, both gram-positive and gram-negative cells would be purple. So all they would have seen in the lab would be gram-positive. They wouldn't have seen gram-negative if they skipped the alcohol step. So D is not our answer either. B is our best answer here. Sorry, I forget it erases things if I scribble on it too much. All right, stop me at any point if you're not understanding one of these or some aspect of these. A patient presents at your clinic with a persistent skin infection that has worsened despite treatment with antibiotics. You'll notice that question 2 and question 3 are going to basically be the same thing, but we're asking a different question at the end of these. Okay. All right, so question 2, we'll set it up. You suspect MRSA. MRSA is methicillin-resistant Staphylococcus aureus. All right. After several tests and culturing procedures, the lab tests are back. The lab reports it's non-motile, it's irregularly clustered. Okay, so non-motile, there's no flagella, right? We can say that. Just making some notes as I go through it. If you guys are answering my questions on my exams, go ahead and do this on your scratch paper or in your mind or whatever. No flagella. It's non-motile, it can't move, so it doesn't have flagella. Irregularly clustered, the word for that is staphylo. Round cells, the word for that is cocci. They grow well on something called mannitol salts agar. Mannitol salts agar has high salt that selects for gram-positive bacteria. It also has a pH indicator that turns from red to yellow if it can break down mannitol. It's telling you right here that mannitol salts agar, and we should infer this from this description here, is both selective, selecting for gram-positives, and against gram-negatives, and it's differential. It's both of those things because we can see a color change that would happen if you broke down the sugar mannitol. So which of the following would be correct regarding this pathogen? Well, here, I'm not asking about selective or differential media, but I could have. What am I asking here? Here, I'm asking if it is a firmicute, if it is peritrichous. Could it be peritrichous? Let's start there. We said it's non-motile. If it's non-motile, it has no flagella. Peritrichous means you have flagella all over you, like this. So it's not this one for sure. Would it test positive for endotoxin? Is it a firmicute? Well, it's gram-positive, which means it has a thick wall that would stain purple in the gram stain. Gram-positives fall into this group, firmicutes. This is a term that you have to know from Unit 1. This is a term you have to know. The firmicutes have the thick cell wall. The gram-negatives are called gracillicutes. The tenericutes lack a wall. And then the mendozicutes are our archaea. Okay, so number 1 is our answer. Let's see why not number 3. Would it test positive for endotoxin? Endotoxin is found only as LPS in gram-negative outer membranes. Since this is a gram-positive, it can't have endotoxin. Okay. Its shape would be described as bacillus. I mean, no, it's cockeye. It's in the name, or it's in the lab report, okay? It's in the name too, but that's what our next question is. We get literally all of the exact same information. This time it's asking us, is the cell shape and the arrangement consistent with our diagnosis? Well, we think it is Staphylococcus aureus. Staphylo, that was our irregular cluster. Coccus was our round cells. So I would say both the shape and the arrangement are consistent. B, C, D, E. On the exam, you will only have true, false, A, B, C, D, or A, B, C, D, E. You will never have more choices than that on the lecture exams. Okay? And only one of them will be correct. It's not multi-choice. But you'll have stuff like this, both, or only the shape, or only the arrangement. Both of them were consistent this time. And there is enough information available. Okay? You will have questions sometimes where this will be the right answer. But it's not this one. Okay? Okay. That was question 3. 1, 2, 3. It started renumbering down here. So this is 4 and this is 5. Okay. The cause of a patient's skin infection is a mystery. Symptoms have worsened upon prescribing wide-spectrum antibiotics. Wide-spectrum antibiotics should have treated most any bacterial infection. A swab of the patient's affected skin was taken, and an initial simple stain procedure was done that indicated large nucleated cells as a source of the infection. Why weren't the antibiotics effective? Antibiotics only treat bacterial infections. So you have to know that too. Wide-spectrum antibiotics should have treated almost any bacterial infection. Antibiotics treat bacterial infections. And here we have the cause of our infection or causative agent has nuclei. This is a eukaryote. They have a skin infection. I'm going to guess it's probably going to be a fungal infection, like some sort of ringworm, but we can't know that for sure with this information. There's not enough information to tell us that it is a fungal infection or that it is specifically ringworm, but I mean, I would guess it probably would be. So why weren't the antibiotics effective? The causative agent was a eukaryote and thus wouldn't be treated using antibiotics. This is the answer. It's a eukaryote. It doesn't make us choose whether it's a fungi or anything. It is definitely a eukaryote though, because we have large nucleated cells as the cause of the infection. Now, some of us might be trying to read too much into this. And they might be like, well, it could have been the patient's cells that were nucleated. I'm giving you that information. I'm telling you that the cause of the infection had large nucleated cells. All that information that you need is there to say that the causative agent was a eukaryote. You do not treat eukaryotic infections with antibiotics. You treat them with appropriate eukaryotic treatments, either antifungals, antiprotozoal, or antihelminthics. All right. The bacteria causing the infection are resistant to the antibiotics prescribed. No, because it's not caused by bacteria, because bacteria don't have nuclei. The patient would likely recover with a stronger dose and longer course of the antibiotics. No, because we're not treating, you shouldn't be treating this with antibiotics because this is a eukaryotic infection. The causative agent was an archaea and thus wouldn't be treated using antibiotics. Now, you might not treat archaea with antibiotics. They might need a special treatment method. However, how do we know that this is not an archaea? Because archaea also do not have a nucleus. So it's not D either. It is A. We're very confident in that. Finally, number 5, a non-smoker patient presents at your clinic with a worsening sore throat. The term for that sore throat, inflammation of your sore throat, is called pharyngitis. Antibiotics, which only treat bacterial infections, I reminded you of that here. I will not remind you of that on the Unit 1 test, but I'm trying to help you with your critical thinking, give you a little information. Antibiotics have proven ineffective. You decide to move forward by sending a swab of your patient's throat to the lab for microscopy. However, microscopy staining finds no visible pathogen present in the sample, and no bacteria or eukaryotic organisms are found to be able to be cultured from the swab sample. What was happening? And more likely than not, you'd have some bacteria from the normal biota, but I'm trying to simplify this for you, okay? Is it A, the antibiotics likely killed the pathogen and the patient is just having lingering symptoms? I don't know, maybe that's not impossible, but is that our best answer? Is it B, the patient is likely infected with a viral pathogen? It's probably B. You're not going to see viruses in a microscope. They're too small. You don't see viruses in a microscope. So it's probably B. All right. The patient is likely imagining their symptoms. Is this possible? Again, it's possible, but our most likely answer so far is B. The patient is probably lying and is actually a smoker. Maybe, but our most likely answer is B still. D, the patient's throat infection is probably caused by an archaea species. No, that we would have seen cells in our culture swab. So it's definitely not E. And our best answer from our other choices is B. It's probably a viral infection. So this is a moment where we have to step back and say, what is the best answer for this situation? And it is B. Okay, so we have made it through. That is everything. Do you guys have any other questions for me outside of this stuff? Make sure that I don't have any chat messages I'm missing. I do not. All right, if that is it, that is my review on chapters 5 and 7. And I went over those critical thinking questions. And that is the level of thinking that I am expecting you on the test. Expecting you to have on the test. About half of the questions will be those level of questions, like what are in those examples there. So expect that. Understand your information well enough that you can apply it in scenarios like what we have in those critical thinking questions. And I would prepare for the whole test to be like that. It won't be all like that. You will have questions that'll be define a term or something like that, or recall a term. And if you prepare for the whole test to be like that, you'll be much better prepared. That's pretty much all I have to say about that. If I don't have any other questions, remember when you guys are studying, you can always email me. Remember to watch the lecture videos. I know I've said it a million times, but there is a lot of information in the lecture videos that you could be missing if you're not watching them. And it might answer a lot of your questions too. So reach out to me if you have any questions during your studying. Um, I will be available on Friday via Zoom, this same link, and I'll probably send out another announcement about it tomorrow from 1 p.m. to 3 p.m. I will be available on Zoom, and I will just be sitting there on Zoom, just like I am here now. I will not be doing anything. I will be sitting there with my computer, probably like grading things or something like that. But if you pop on, come pop on and ask questions. And if anybody shows up and there's any relevant information that I think other people could gain from the discussions, then I might post it in like a discussion post or something. So don't think you're going to miss anything with that if that happens. Again, it's just meant to be office hours. So if you have any questions, please, feel like you're willing to come and have those discussions or whatever with me. All right. Any other questions?
No, thanks for your time.
Absolutely. So happy studying. Contact me if you need to. Otherwise, Sunday at 4 p.m., we'll have our gaming session. I hope to see you all there. Thanks a lot, everybody. Have a good evening.
Thank you.